Okay, in this lesson, we'll take a look at a brain imaging technique called functional magnetic resonance imaging, fMRI. You've probably seen people being inserted into these giant magnets. So this is the MRI machine, and it is able to generate computer images not only of the brain itself, but also of the real-time activity of different brain areas. So it's a very powerful technique for studying the brain. This will be a typical product of an MRI study. So here we see the brain, and the little color patches here would indicate regions of the brain that show elevated levels of activity. Right, so usually the brighter the colors, the more activity. In this case, it's the back of the brain that's showing activity, and so we know that to be the visual cortex. So in this task, the person was probably inserted into the machine, and they were asked to observe something on a little computer screen. So it was a visual task, and the MRI picked up the signal. So the back of the brain was showed elevated activity. Now, to understand how the MRI system works, first let's do a little background. You'll recall that we've looked at the cortex here for a little bit, and you know that there are these numbers associated with different regions of the cortex, and that's for historical reasons. Some of the early pioneers who were mapping out the cortex were doing studies of the cellular organization of these different areas, and they found that there were differences across the surface of the cortex. But any particular region that had a similar cellular architecture was designated with one particular number, right? So area 17 we happen to know is primary visual cortex, area 41 auditory cortex, area 312 somatosensory cortex, area 4 here would be motor cortex. And as we'll see, these areas have different functions, right? Principally due to their input-output relations. So the inputs to area 17 will be different than the inputs to area 41. Of course, area 17 is going to get inputs from the eyes, and area 41 inputs from the ears. But more on that later. If we took a look at what the cells of the cortex look like under the microscope, you'd see something like this, a forest of cells. So the cells have been stained here. The dark blobs here are the cell bodies, and you can see that nerve cells, brain cells, have a lot of extensions, branches off of them. Here we see a single brain cell body in the middle here, and this extensive network of branches called dendrites, and those are the receiving areas of the nerve cells. So hundreds or thousands of different neurons might make connections with these dendrites. And then coming out of the cell body is the output cable, you might say, called the axon. And the axon will relay an electrical signal to some other nerve cell in the brain, or perhaps a muscle. Here we see a bipolar neuron, so here's the cell body, 2 clusters of dendrites, and here is the axon right here, tiny little one. It would emanate out of the cell body, and they often branch, so they can go to multiple targets, but that would be the output cable of the cell. So every region of the cortex then is composed of a whole sort of network of cortical cells. Here's a part of the brain that we are a little bit familiar with now. Here's the temporal lobe here, and underneath the temporal lobe is the hippocampus. Now it's a cross-section of the hippocampus. And you'll notice, though, that the temporal lobe, there's sort of some dark matter here, and then there's lighter matter in here, right? And that corresponds to the cell bodies are located out here in the surface of the cortex, and then the axons, or the nerve fibers, the output cables, constitute the white matter. We can see the same thing down here on this picture here. Again, the hippocampus is right here, but here the cell bodies have been stained a purplish color. But you can now clearly see the cortex, the outer surface of the cortex is where the cell bodies are. And all this material in here, all this stuff in here is white matter. So those would be the axons connecting brain cells with each other. All right, if we zoom in on the hippocampus, here we see a lower power magnification of the rat hippocampus, and we can begin to see individual cells in a higher power magnification. And now we can start to see individual hippocampal cells. Now, brain cells are electrically excitable so that we can actually record the electrical activity of populations of cells or individual neurons. This would be a microelectrode, and you're actually seeing one neuron here. The electrical nature of brain cells has to do with the fact that they move charged particles across their membranes, and when a charged particle is on the move, that's an electrical current. So neurons are electrically excitable because they are propagating these electrical disturbances down their membranes. And that disturbance is the movement of charged particles like sodium ions and potassium ions and chloride ions through protein channels that are situated in the membranes of the cell. So when you're doing electrical recording, you're just trying to listen in on the faint electrical activity of individual cells or whole populations of cells. Of course, if you can record from a cell, you can also stimulate it as well. And so there are various kinds of studies that either make use of recording or stimulating electrodes. Okay, so let's now get back to magnetic resonance imaging. And for that, we're going to watch a little clip. Okay, so here's our individual inserted into a giant magnet, and the strong magnetic field here is going to align the proton, the hydrogen ion nucleus is a proton, and those things are spinning, and when they're in a strong magnetic field, they're going to align their sort of spinning axis with the magnetic field. But let's take a look at what they say here.
MRI uses a magnetic field to orient the position of certain nuclei in the body. The primary nuclei used for MRI imaging is in the hydrogen atom, called a proton. As you know, the body is made up of approximately 75% water. Water is 2 parts hydrogen, making the nucleus of the hydrogen atom, the proton, the most abundant in the human body. A radio wave is then used to excite the proton, which resonate and emit varying signals, which are received, digitized, and displayed as images. Different signals emitted by different body tissues are then used to differentiate individual anatomy. When tissue is damaged, its composition changes and gives off an uncharacteristic signal. So when displayed, that damaged tissue is distinguishable from surrounding healthy tissue.
Okay, not the most detailed explanation of how the MRI system works, but it is somewhat complicated. But for our purposes, we just want to know that the technique allows you to take a pretty nice picture of the anatomy, the tissue structure of the body, including the brain. And so different tissues have different characteristic signals, right? So because they have different densities of water, they have different thermodynamic properties, and so they will show up on an MRI image as distinct regions. Let's take a look. This is something you might see then, for example, in an MRI. So you can definitely see the difference between, say, brain. So here's the brain up here. We've got the skull here. We've got the sinuses. We've got the spinal cord. We've got vertebrae and the tongue and so on. So different types of tissue end up giving off different characteristic signals, and that's what allows you to see the detail in the image. But this picture here is only an MRI image. It is not a functional image, so we don't know what parts of the brain are especially active in a picture like this. So this is just MRI imaging of the brain. It's giving you a picture of the anatomy of the brain, and if somebody had a brain tumor, for example, this would be useful for determining the location of the tumor. But this is not the technique that will allow you to look at real-time brain activity. For that, we have to discuss the technique called functional MRI imaging. Okay, so to understand the functional MRI, we have to know some basic biology. The basic biology is that brain cells, they absorb glucose and oxygen from the blood. Glucose is the main energy molecule for the cells in our body. So brain cells absorb glucose, and then they break glucose down to extract energy. And that, of course, powers the electrical and chemical nature of brain cells. Oxygen is needed for that glucose metabolism. So oxygen is, of course, delivered by red blood cells. So all brain cells need a plentiful supply of blood. Blood is carrying critical nutrients for brain activity. And when brain cells are highly active, when their activity increases, then it stands to reason they do need more nutrients, right? So they will be absorbing more oxygen and more glucose from the blood. So let's take a look in this picture here. Here we see individual neurons growing on the surface of a petri dish. Now, to keep them alive, you would have to supply them with nutrients like sugars, and there would have to be plenty of oxygen and dissolved in the solution there. Of course, in a real brain, those nutrients are delivered by the blood. So here we see a brain where the blood supply has been indicated by injecting a chemical that then can be sort of photographed. And here we see, a major artery branching off into sub units, so smaller arteries delivering nutrients to the different parts of the brain. Here's another sort of picture like that. So the entire brain then is bathed in the blood supply, right? No nerve cell is very far away from a capillary. Now, a really important bit of biology here to understand functional MRI is that local blood vessels will increase blood flow to those areas of the brain that are showing greater levels of activity. So in other words, when a certain part of the brain becomes more active, the local blood flow increases to that area. And that happens in your body too. For example, when you use different muscles. If you start running a lot, the muscles of your leg are going to start to recruit or start to need more nutrients from the blood, and blood flow will increase to those muscles that are in use. Here we see the surface of the brain, and we see some arteries, right, servicing every nook and cranny of that brain. So again, the idea is if this area down here, they've just put a little number there, 14, if that area is active for some particular task that the person is doing, the local blood vessels in that area will start to increase the blood flow to that area. And that's going to be the key to this functional MRI technique. Okay, so here's the key step here. It turns out that blood that's carrying oxygen has different magnetic properties than blood without oxygen. And so therefore, the brain areas receiving increased blood flow can be measured, right? And that is, remember, the MRI is measuring these signals coming from those hydrogen nuclei that are emanating their little signals when they're placed in a large magnetic field and radio waves are being sent at them. Those little signals will depend on how much blood with oxygen is going to a particular part of the brain. So the functional MRI measures blood flow changes, right? And that's being used as a proxy for brain cell activity. So the technique is not actually measuring brain cell activity directly, but it's measuring changes in blood flow that are the result of changes in brain cell activity. If we take a look at a little diagram of how this would work, here we see a blood vessel here on the left, and here we see some neurons here, and they are situated so as to absorb nutrients from the blood. Okay, the red here would be oxyhemoglobin, so this would be oxygen-rich blood. So here comes the oxygen-rich blood, and these cells then are going to be absorbing the oxygen. And so as the blood passes by this group of cells here, the oxygen will be depleted. So this is deoxyhemoglobin, right? So the idea is that the magnetic properties of oxygen-rich blood are different from the magnetic properties of oxygen-poor blood. And so if these cells become highly active, they're going to be absorbing more nutrients from the blood. The blood vessels nearby respond by increasing the blood flow to those neurons. And so then the MRI device is going to be picking up a signal, a change in the magnetic properties of that region, because the blood flow has now increased, and there's a difference between the magnetic properties of the oxygen-rich and the oxygen-poor blood. In terms of looking at sort of the signal that you might measure from this, up here we have, say, a record of the brain cells in the area, and the little hatch marks here would be indicating that the brain cell is electrically excited. So let's say, so the brain cells in that region, they're getting real excited. They're making a lot of electrical signals, right? So that means that their energy demands are going to increase, and they're going to be absorbing a lot of oxygen. Here goes the oxygen out of the blood. Oxygen and glucose, so these are highly active cells. They're going to be absorbing, so we're going to get a temporary decrease in the oxygenation of that blood here, right? But then the blood vessels are going to respond by increasing the amount of blood flow to the region, so then you'll get an increase in the oxygen content of the blood there. So we get the signal then goes up, and as that oxygen gets delivered, it's going to be absorbed, of course, by the active brain cells. But then when the brain cells quiet down, right, then the blood vessels will decrease the blood flow to that area, and you'll get a return to the baseline. So when a part of the brain is temporarily active, you're going to get this increase in this signal that is tracking the increase in the activity of the brain cells in the area. It's called a BOLD signal, for blood oxygen level dependent signal. And again, here's a little picture of how that works. In the resting state, there's a certain amount of oxygen utilization, but when neurons are highly excited, when those brain cells are doing some heavy computations, there will be a greater need for oxygen. Blood vessels respond by increasing blood flow, and you will get a change in the signal as registered by the functional magnetic imaging system.